All right. Welcome to the CTSI seminar. This is for the regenerative medicine theme, and I have to say this is the most full I've seen this room since the pandemic started. So we've got a big draw, and thank you all for coming out today. And we've got a lot of people on Zoom as well. So this is a very exciting introduction that I get to make because the person that we have speaking today actually trained me in the lab, taught me how to make virus, how to dissect mice, and do flow cytometry, which is still my favorite technique. So I'm indebted to you for the time and energy you put into training another scientist, and I know you've just continued that on in your career. And so I'm very excited to introduce Dr. Devon Lawson. She's an associate professor at UC Irvine. Devon has made her way through the University of California system in her career. So she started at UC Davis, studying biology and genetics. She came down to UCLA. She did her PhD with Owen Witte, studying prostate stem cells. She made enormous contributions to the isolation of stem cells in the prostate and studying their role in prostate cancer. So I looked this up this morning. She had 3 gigantic publications in graduate school, and collectively they have nearly 1,300 references. So she's made a gigantic impact in that field. The SCA1 paper having over 500 citations, which is very impressive. After graduate school, she went up the coast to UCSF, where she did a postdoctoral fellowship with Dr. Zena Werb. And she developed new technologies to study the early stages of metastasis using patient-derived xenograft models and incorporating single-cell sequencing to identify a unique subset of cells with stem cell-like properties in breast cancer that contribute to early seeding of metastatic sites. She was then recruited to UC Irvine in the Department of Physiology and Biophysics. She's also a member of the Chow Family Comprehensive Cancer Center and the Gross Stem Cell Research Center. At UCI, she's been really an expert in single cell technologies with breast cancer. She's been part of these major consortia. She's part of the Human Cell Atlas project, which is basically an international collaboration to sequence all the cells in human tissues. And this has been a gigantic effort. She's really been a early adopter of this technology and made huge impacts. She's received so many awards supported from DOD, NIH, American Cancer Society, V Scholar, Susan G. Komen Foundation. She was also awarded the ACS Trailblazer Award. And so I'm very excited to welcome her back to UCLA and to hear what she has to say. Welcome, Devon.
Great. Well, that was fun. Thank you, Andrew, for that introduction. And it's really great to be back here. I don't think I've been here for 10 years almost. I think I came to visit Owen maybe in 2010 or something in the lab. And I have to say, when I was driving up Westwood, it was really quite like a little bit like coming home after a long trip away. How many grad students do we have in the audience? I have some. So I did my graduate work here in Owen Witte's lab, and just like Andrew was saying, really was trained to do science. You feel a little bit like you grow up in grad school. I spent most of my 20s here, grew up as a person and also as a scientist. Most of your training really in how to do science happens in graduate school. In postdoc years, you're more trying to like make the big discovery, right? Using what you've learned. But I definitely feel like this is kind of where I grew up. So it's great to be back. And today I'm going to tell you a little bit about our work down at UC Irvine studying breast cancer metastasis. And here I have actually a picture from the Human Cell Atlas project of the human breast, where we've been using a single cell spatial technology called Codex to do multiplex immunofluorescence analysis and really try and localize the many different cell types and cell states in the human breast. Let's see. Go forward. There we go. Thank you. So In my lab at UCI, we think a lot about intratumor heterogeneity in the context of breast cancer metastasis. And we study this problem from two sort of main angles. And so you can see that kind of represented here in this schematic. So in trying to understand what kinds of cancer cells can metastasize and what signals from the microenvironment either inhibit them or promote them in doing so. Basically, we think about the system in this way. So in thinking about the cancer cells here, which are shown in blue, there are a lot of different ways in which cancer cells can be heterogeneous. They can have different genotypes. They can have different differentiation states, different metabolic states. But the microenvironment can also be very heterogeneous. So for example, cells can differ in their access to the blood supply, angiogenesis. They can also, tumors can also be different with respect to what kind of immune cell infiltrate they tend to be exposed to, whether it be anti-tumor immune cells or pro-tumorigenic immune cells, something that we think a lot about in the lab. So today I'm going to tell you two studies that we've been working on for the last 5 or 6 years in the lab. The first focus is on the tumor cells themselves, trying to understand cell intrinsic mechanisms that allow cancer cells to metastasize. And in that study, we found that actually there are these really interesting metabolic promoters of metastasis to the lung. And then in the second half of the talk, I'm going to tell you a little bit about our work studying the tumor microenvironment, trying to understand, again, how immune cells in the microenvironment regulate metastasis. And in that study, it's in the context of actually brain metastasis. And as Andrew said, in my lab, we've really focused very heavily on using single cell technologies. So when you think of tumor heterogeneity, right, you think of all these different kinds of cells, and also not only how they behave individually, but also how they behave collectively in a system. And so we've used these single cell technologies to really get at fundamental biology of what kinds of cancer cells can metastasize and what kinds of different cells represent or reside in their microenvironment. And so at UCSF, when I was a postdoc, as Andrew mentioned, we started really using these pioneering sort of early single cell technologies where we were just doing multiplex qPCR of single cells with this old biomark system, which actually worked quite well. But at UCI, we've moved on and moved forward really to using, sort of your more workhorse. You guys are probably more familiar with the droplet-based methods, for example, by 10x Genomics. And then in more recent years, through our work on the Human Cell Atlas project, we've moved a lot into spatial work, again, trying to understand how these cells function in their actual native microenvironment. So I'll show you a little bit of work that we've done on the codex. And unfortunately, don't have a lot of time to show you work on the Cell Atlas project, but we've done all kinds of other spatial technologies, from Visium to Murfish to in situ hybridization. And I'd be happy to tell you more about our experiences with those technologies sort of offline, if you're interested. Okay, so getting to the first study that I wanted to tell you a little bit about. This study, again, was focused very particularly on investigating cell intrinsic properties within the cancer cells themselves that enable them to metastasize. And we always think of this paradigm in the metastasis field about how cancer cells metastasize, that they have to be able to go through all of these different steps in what we call the metastatic cascade in order to actually seed in a new tissue and create a new tumor. So again, going back to this idea of tumor heterogeneity, we think of tumors again as this sort of conglomerate of different kinds of cells that have both different phenotypes as well as different genotypes and functions. And the way we think about this in our lab is that it takes a very special kind of cell, which has a unique combination of both phenotypic and genotypic properties that can actually get out of the primary tumor, survive within the bloodstream, extravasate at a metastatic site, and then survive and seed in a completely new sort of soil or tissue, in this case, in this study, in the lung. And then furthermore, after even being able to survive and seed, being able to produce micro and macro metastasis. So in this study, in these first studies, we're really trying to track down which of these cells in the primary tumor, what is the phenotype of this cell that is actually able to do this? So during my postdoc, really the first step was being able to isolate metastatic cells when they're seeding in metastatic tissues. And this actually sounds kind of trivial, but it actually took several years of work during the first years of my postdoc. And sort of in a nutshell, what we were doing was trying to use these patient-derived xenograft models, which we hoped at the time were a more authentic model of human breast cancer metastasis than some of the cell line-based models that had been used historically. And so for those of you who aren't familiar, PDX models, you take primary tumor samples, transplant them into the breast of mice, and then we look to see whether they can metastasize to distal organs. And the way that I developed a method to fish them back out, sort of during the seeding process, you can see these little micromets here forming in the lung of these PDX mice, was to develop this flow cytometry assay that relied on two sort of fundamental principles. Essentially, that human cells and mouse cells are distinguishable with different markers, right? So in this assay, what we ended up using was a mouse MHC1 antibody. So basically, all cells in the mouse express MHC1 and the antibody is species specific. So you can see in the lung, most all of the cells are MHC1 positive. Tumor cells from the human, however, don't express mouse MHC1. And it turns out they all express this sodium potassium ATPase called CD298, just essential for function, basic cellular function. And this is not expressed in mice. And so as you can see in the tumor, for control, pretty much all the tumor cells are CD298 positive, and there are some infiltrating mouse immune and fibroblasts as well. And this has all been validated, and I won't show you all the validation data, but it turned out to be a really important sort of principle and workhorse for my postdoctoral project and also many of the projects in the lab. And in my first sort of experiments in Zina Werb's lab were really inspired by work that I had done here, which Andrew mentioned and alluded to, really in Owen Whitty's lab, where we were really interested in sort of the early and mid 2000s, trying to understand cellular differentiation in the prostate. So you can see, you guys know this better than I do now. There are these different cell types in the prostate, basal cells and luminal cells. There are stem cells and progenitor cells embedded within them, probably still not entirely understood, neither in the breast, where exactly or what exactly they do. But we published a series of papers trying to understand cellular differentiation and isolate stem cells from the prostate. And then furthermore, trying to understand how these different kinds of cells participate in tumor initiation in the prostate. It turns out the breast is actually fairly similarly organized. So you can see here a cross section of a duct here from the breast, has a very similar composition of basal cells around the outside and these luminal cells sort of in the interior. And again, stem and progenitor cells embedded within these layers. That's something we've been trying to understand through our work on the Human Cell Atlas project. But in my first work in Zina Werb's lab at UCSF, what I really wanted to understand was whether metastatic cells had a unique differentiation state relative to the bulk of primary tumor cells. And so I designed my single cell experiments to get at this very specific question. And at this time, we were using these microfluidics-based technologies for doing multiplex qPCR. So you had to actually design qPCR assays to address this question. And so in this study, I designed my assay, my qPCR panel, essentially to look at stem cell and differentiation genes. And so to make a long story short, we used 3 different PDX models, 3 different patients, compared primary tumor cells and metastatic cells from the lung and lymph node using our flow cytometry assay, and looked to see what their gene expression patterns are. And to sum up like 5 or 6 years of work in one slide, so I don't go too far into published work. In a nutshell, what we found was that basically the metastatic cells overwhelmingly upregulate or show increased expression of a whole slew of basal and stem cell associated genes. So you can see here our primary tumor average expression is basically this 0 line. And we found all these genes, LGR5, BMI1, BCL2, Notch4, keratin5, were higher expressed in the metastatic cells relative to the primary tumor cells. And conversely, genes that we usually associate with luminal differentiation like EMP1 and CD24, MUC1, things like that were lower in the metastatic cells. Even some of these pluripotency genes like SOX2 and OCT4 were higher, much higher in the metastatic cells than the primary tumor cells. So from this, we were able to conclude at this very low resolution at this time, although we were able to publish it very nicely at the time since it was state of the art at the time, that it does seem in order to metastasize in these models, the cancer cells really need to have some stem cell related gene expression programs. But going forward at UCI, we wanted to look past, a few genes and look at really the whole transcriptome and see if we could learn more about what kinds of cancer cells are able to metastasize and what programs, enable them to do so. And so my first graduate student here, Ryan Davis, who's actually now a scientist at Caribou Biosciences in Berkeley, basically used this same pipeline to compare gene expression patterns in primary tumor and metastatic cells. This time using a whole transcriptome method, which actually predates the 10x genomics droplet-based method. We're actually doing whole full-length RNA sequencing in single cells at this time, which was important also because we were looking at very rare cells in these micrometastatic lesions of the lung. And you can see it uses this unique chemistry here called SMART-seq2, where it was actually a oligo DT priming method, as well as a template switching oligo and enzyme that enabled us to capture the cDNA, or the RNA, make it into cDNA, and then amplify it up so that we could actually detect gene expression differences in our cells. So the first thing that Ryan did when I guess in this study, we also, very much like the study I just showed you, we looked at 3 different PDX models, which you can see here, patient 1, patient 2, and patient 10. And the first thing we just looked at was transcriptional diversity. So is there any heterogeneity at all, even in these patient tumors? And you can see, those of you who are familiar with looking at single cell work, obviously this predates the millions of cells, pipelines that we actually are doing now. But even with these smaller numbers of cells in the SMART-seq method, we were able to see some heterogeneity within the tumors. And you can see here the marker gene expression heat maps show that there were these discrete subpopulations of cancer cells within these 3 PDX models. It bears noting for those of you who have done this kind of stuff, the tumors, when we assay them, are never, the subpopulations are never as discrete as we observe in normal tissues. Cancer cells are much more plastic. There's a lot more dynamic behavior and gene expression, and so they tend to cluster closer to one another. They're less different than normal cells, but you can see that we do see these discrete gene expression programs acting in different cell populations. But ultimately, what we were really interested in, again, was whether the metastatic cells in the lung have a unique program that hopefully, enables them to metastasize relative to primary tumor cells. So we did this pseudo bulking method where basically we went to our single cell data and collapsed it into pseudo bulk analysis and looked at our primary tumor cells and our micrometastatic cells and looked for conserved markers that were upregulated in all 3 of our models in the metastatic sites. And ultimately, what Ryan found was this signature of 330 genes across the transcriptome, which were differentially expressed in all 3 models in the metastatic cells compared to the primary tumor. And here's just sort of the top 20 or so of these differentially expressed genes. And we saw some things in common actually with our qPCR assay, some of these basal genes like keratin 14, for example. But we also saw some genes that which have unknown function. For example, PHLDA2 is one we followed up on. It's a very interesting maternally imprinted gene, which turns out to be associated with very poor survival in breast cancer patients. And then all these genes here, for example, that were upregulated or they were higher in the primary tumor cells than the metastatic cells. And actually, when we created a gene signature to look to see whether these were predictive of poor survival, we actually saw very significant differences in our breast cancer, basal-like breast cancer patients, where patients who express high levels of this gene set, you could see were basically outcomes were much worse with a hazard ratio of 2.7, which actually is pretty significant. So there's definitely biological and clinical relevance of this gene set that we were looking at. But going forward to try and look beyond just individual genes and gene sets, we did some gene ontology analysis of our 330 gene signature. And overwhelmingly, the top hit that we observed was a metabolic change. So it was very clear that our metastatic cells were upregulating genes associated with mitochondrial oxidative phosphorylation. So you see all these hits: respiratory electron transport chain, ATP metabolic process, OXPHOS, were higher in the in the metastatic cells. Whereas in the primary tumor cells, we were seeing many more genes associated with canonical glycolysis. And at this time, we were not experts in metabolism in any way, shape, or form. And so for those of you who also aren't, you probably have heard of the Warburg effect and this concept that tumor cells, or highly proliferative cells, often prefer to use aerobic glycolysis to create cellular components and enable them to proliferate rapidly. So that was sort of a surprise to us, that our metastatic cells seem to prefer a different type of metabolism. It turns out that normal cells, particularly also stem cells often, depending on the system, also preferentially utilize oxidative phosphorylation over Warburg metabolism. We also did some gene scoring, basically for all the different types of metabolic pathways, just as a sanity check to see whether indeed this was true. And when we did gene scoring for whole lists of genes associated with the different metabolic pathways, the top hit, again, the top scoring pathway was glycolysis in our primary tumor samples, and OXPHOS, as well as some actually amino acid pyrimidine and purine metabolism as well in the micrometastatic cells. Hopefully to further convince you beyond Go term analysis, any of you guys who do computational work know that it's easy to like get a bunch of Go terms and none of them actually make sense or are true. But when we actually looked at the genes overexpressed in our data set, we did in fact see a whole set of genes associated with oxidative phosphorylation. You see these mitochondrial complexes here, for example, and some of the transport proteins were indeed upregulated in the micrometastatic cells. Whereas we saw a number of glycolysis enzymes, for example, ALDO-A, to basically upregulated in our primary tumor cells. So it was a good validation that our analyses actually were making sense here. We also went on to do some functional validations. That's always the next step, right? When you're doing gene expression analyses. So we looked to see how can we actually see whether our metastatic cells are performing more mitochondrial respiration. And so what we did was we utilized this dye, which essentially senses differences in charge across the mitochondrial membrane. So when the cell or the mitochondria is more actively respiring, you get an accumulation of protons, and that causes increased fluorescence when you add this dye. So what we did was we compared primary tumor cells and micrometastatic cells by flow cytometry for fluorescence with this dye. And across the animals that we looked at, we also saw higher levels of mitochondrial membrane potential, essentially, in the metastatic cells compared to the primary tumor cells using this assay. We also tried our hand at some steady state metabolomics. This was a really interesting collaboration that we started with Jason Locasale's lab at Duke University. Again, for those of you who've done any metabolomics, trying to do it with small cell numbers, for example, micrometastatic cells, is almost impossible, but they thought too, why not give it a shot? So we tried our hand at comparing metabolite levels essentially in primary tumor cells compared to metastatic cells that we isolated, again, using our flow cytometry assay. Did cold methanol extraction to extract the metabolites and then ran them on his LC-HRMS instrument at Duke. We weren't able to detect all the metabolites, but we were able to detect the most abundant metabolites in these cell populations. I think we detected maybe 150 or 160 metabolites across the data set. And there are no metabolites really directly associated with OXPHOS, but there are a lot of intermediates that can feed into OXPHOS. So what we saw basically were some of these intermediates at higher levels in our metastatic cells, the highest being this fatty acid metabolism intermediate called palmitocarnitine, which you can see here, was the highest in our metastatic cells. But we also saw some TCA cycle intermediates, as well as like glutamine and glutamate, which also, are important in cancer cell metabolism and feed into the TCA cycle. So now we're moving forward and trying to do some more tracing experiments to see if we can confirm some of this and look beyond at how nutrients are being metabolized in metastatic cells compared to primary tumor cells. We also did some functional experiments. So again, thinking about mitochondrial respiration, our hypothesis was that the cells, for some reason, prefer this type of metabolism to enable them to metastasize. But the alternative hypothesis, of course, is that it's just like there's more oxygen in the lung, for example, right? Or in the blood as they're metabolizing. And so it's not necessarily functionally important at all. It's just a side effect of them being able to use oxygen. So we did some functional experiments to see whether indeed the cells need a lot of OXPHOS in order to metastasize. And what we did was we used this compound called oligomycin, which essentially blocks the compound 5, the ATP synthase in the electron transport chain, and shuts down or slows down respiration. We tried this compound in 2 different cell lines, so a human breast cancer cell line and a mouse breast cancer cell line, and we treated the cells in culture for, I think, in 24 hours. And then we harvested the cells and injected them intravenously into mice and looked to see whether the treated cells had less or more metastasis. And of course, before we injected them, we looked to see whether, in fact, we were able to modulate respiration. And you could see that the treated cells did have lower respiration by seahorse analysis. Importantly, we wanted to make sure we didn't kill the cells by slowing down their ability to respire. So we first injected the cells just into the memory fat pad, and we saw that the tumor still grew in relatively similar sizes, even though we had treated them with oligomycin overnight. And then most importantly, using our flow cytometry assay, we looked in the lungs of these mice, I think a few weeks later, to see whether there was a lower metastatic burden after oligomycin treatment, even for a short term. And what we did find was indeed less metastatic cells in the treated mice in both the MDA 231 and the 421 injection experiments. So this suggested to us that, yes, in fact, these cells seem to need to be able to use OXPHOS. Even for short periods, reducing it may affect their ability to survive, for example, in the bloodstream or seed in the lung in that first initial period after treatment and injection into the mice. We also have done some experiments, some preliminary experiments, looking to see whether these cells are observable in the primary tumor. So like you can imagine a couple of different scenarios, right? Like there are cells in the primary tumor which use glycolysis and cells that use more mitochondrial respiration. And it's these guys that are the ones that metastasize, right? That was our hypothesis. Alternative hypothesis could be that they upregulate OXPHOS later on, after they leave the primary tumor in response to nutrient availability or something in sites outside the tumor. So in order to answer this question and distinguish between these 2 principles, a postdoc in the lab, Dennis Ma, who's actually now a senior scientist at Calico in the Bay Area, devised this assay again using the dye to sort cells that were high for mitochondrial membrane potential versus low for mitochondrial membrane potential, and look to see whether there's any difference in their ability to metastasize. Again, using primary tumor cells from these PDX mice. And you can see here the flow plots. There were these cells that were high for TMRM and low for TMRM. About 5% or so in this particular model were high versus low. And what he found was more seeding, essentially, in the cells that were using more OXPHOS, more seeding in the lung. And he was able to measure that both by whole mount fluorescence. These cells were GFP positive, as well as using our flow cytometry assay here, which you can see quantified in the cohort of mice. So suggesting again that these cells may already be pre-existing in the primary tumor, and that it's not a response to the microenvironment, but rather something already early on that enables them to escape the primary tumor. And then a couple of last things that I wanted to mention. The next steps in this project really are, we're trying to figure out, well, first I wanted to mention that we're not the only ones in the field that have actually observed similar phenomenons. This paper here, published by the Kaluri Lab in about 2016, I think, found something similar, where they were looking at circulating tumor cells in a breast cancer injection model. And they found that the circulating tumor cells, when assayed by RNA sequencing, also seemed to upregulate OXPHOS in the circulation. And that was in contrast to the primary tumor, as well as macrometastatic tumors that they observed in the lung. And you can see that represented here in their RNA sequencing data. And they found actually that there was this mitochondrial biogenesis gene, PGC1 alpha, that actually mediated this change and was a driver of metastasis in their models. So definitely there's mounting evidence in the field that OXPHOS does seem to be important, particularly for the metastatic processes, cancer cells. But as I was saying, the next steps in this project, what we've been really thinking a lot about is why. So what is it that respiration provides to the cell that makes it more able to metastasize, or that helps it metastasize? And there are a lot of different things you could think of. One being ATP. Obviously, OXPHOS makes a ton of ATP, makes a lot more ATP than glycolysis, for example, per input molecule. OXPHOS also makes a lot of ROS. And, ROS is like that Goldilocks molecule, right? Where you have too much, it's bad, and you have too little, it's bad. But if you have the right amount, it can actually induce very potent physiological responses in cells. And so we've been thinking first about ROS, although we thought about other things. And so we use this compound called rotenone in preliminary experiments, where when you treat cells with rotenone, it actually blocks complex 1 and preventing, transfer of electrons down the chain, essentially accumulation of electrons and ROS in the cell. So these cells make more ROS. There's actually a graduate student in the lab, Paige Hallis, who'd been working on this for a little while. And in some of her experiments, although not all of her experiments, it's kind of an interesting phenomenon that we're trying to understand. But in some of her experiments, it seems in some cancer cell lines, increasing ROS does, when we inject them intravenously into the bloodstream, it increases their ability to metastasize. So there may be, ROS may be part of the story. And why ROS? Well, ROS does a lot of things, like I just said. OXPHOS makes a lot of ROS. We know already from other prior work, we know a lot about ROS and what it does. Definitely can cause angiogenesis, invasion and migration, resistance to anoicus, a lot of things that cancer cells may want to be able to do as they're metastasizing. But as I said, there are a lot of other things, other mechanisms that we've been thinking about in this project. One is this really interesting paper that was published a couple of years ago. There's a postdoc in the lab who's very interested in this sort of symbiotic relationship that they observed in primary tumors of cancer cells that use glycolysis versus cancer cells that use more OXPHOS. And it turns out in this paper, what they found was the Warburging cancer cells make a lot of lactate. That's a byproduct, right, of Warburg metabolism. Lactate is actually then uptake by neighboring cells that have access to oxygen and can do a lot of OXPHOS with that lactate. And that they found that those cells are the cells that actually are more prone to metastasis than the lactate producing cells. It's really cool, cool symbiosis mechanism, I think, that they found we're very interested in. And then lastly, another phenomenon that could be why OXPHOS is important. There were these whole series of papers published by David Sabatini, Matt Vanderheiden, showing that the main function of respiration, or a main function of respiration for proliferating cells, is to produce aspartate. So you need aspartate in order to proliferate. And OXPHOS is one of the main ways in which you can make aspartate in the cell. And so it could be that simply these cells need to make this, and they make more OXPHOS in order to make it. So we've been thinking about that as well. Most of this work wasn't done in the metastatic setting, although there's this nice paper by the Henin lab that showed that this is very important also in metastasis. So that's basically where we're at with this study. So just a few conclusions. We found in these studies that PDX tumors have different subpopulations of cells. We found a unique gene signature that seems to be acting in metastatic cells using single cell analysis. OXPHOS is a main program. Cells with higher levels of OXPHOS display increased metastatic potential. ROS, potentially other mechanisms, may be part of the how the OXPHOS helps the cells metastasize. So with that, I'm going to go on and tell you in the last few minutes, I think we have a few minutes left, about a second study that's actually all unpublished. We haven't published the other. We're trying really hard to get this paper finished up and out. It's a really cool new study, also pioneered by one of my first graduate students in the lab and taken over by many additional graduate students since that time. And in this project, in contrast to the study that I just showed you, where we're looking at the cancer cells themselves and how they promote and what in them promotes metastasis, here we're looking at microenvironment. Okay, so we're looking at immune cells and how they basically control and regulate breast cancer metastasis to the brain. So, as I'm sure many of you can imagine, unfortunately, brain metastasis is something that we started studying 5 or 6 years ago in the lab because it represents a really horrible clinical unmet need. Brain metastasis in breast cancer patients causes rapid fatality, in contrast to primary disease, which is actually quite controllable for breast cancer patients. 30 to 40% of patients with triple negative breast cancer actually develop brain metastasis, quite a lot of patients. That translates into 150,000 new cases per year compared to many fewer for brain cancers, but actually most work still is done on brain cancer itself. As you probably are also aware, most therapies can't cross the blood-brain barrier, leaving us with almost nothing to treat these patients with. And so, as I mentioned at the beginning, median survival is really awful for these patients, about 5 months or so. So we're really trying to make an impact and understand this stage of metastatic disease better. So we started this project. It's very clear that sort of a defining feature of brain metastasis, really all brain metastasis, including breast, is that they're extensively infiltrated with tumor-associated macrophages. So you can see that here, these IBA1 positive cells in red are just, sometimes they comprise 50–60% of the tumor. And it's interesting because the brain has its own resident macrophage population. They're called microglia, and you can see them here. They have this unique ramified morphology. And we know that that's pretty much all the macrophages in the brain in sort of normal homeostasis. In disease, it becomes very difficult to determine what kind of macrophages these are and where they came from, because in inflammatory conditions, microglia look more like peripheral macrophages. And when peripheral macrophages come into the brain, they start to look more like microglia. So it's anyone's guess, really, what these cells are and what they're doing. So we decided to try and tackle this. And in the field, in brain cancer, particularly, it had been published over and over again that these cells are immune suppressive and pro-tumorigenic. That is definitely sort of the dogma in this field. And not surprising, we know TAMs often are immune suppressive, and there are a lot of clinical trials out there trying to target them and make them go away. So we started to look into this in the brain metastasis setting. And as I just mentioned, it's very difficult to discriminate all these different kinds of macrophages from the periphery and the CNS. So we decided to tackle this with a single cell RNA sequencing approach and see whether we could use whole gene signatures to figure out what kind of cells these are and what they're doing in these brain mets. So as I mentioned before, Katrina Evans, who's actually at Akoya now, she took on this project early on. It was really, I was more interested in the brain cancer cells. She's more interested in the immune cells. So we definitely credit her for getting this project launched and going. And we turned to a mouse model to try and understand what these cells are doing. This is a human cell line injected into an immune deficient animal. Brain metastasis follows a pretty standard course. You can see the mets here in the whole mountain of the brain. At day 7, you already see these IBA1 positive microglia, most likely at this time point, surrounding the cancer cells in green. And that progresses and accumulates at day 14. And then by day 28, when you have more macrometastatic tumors, you start to see tons of these cells, again, just like in the patients. So it's a very good model of TAM infiltration in the mouse. Second graduate student, Kerrigan Blake, who actually was a computational student. She's actually now a scientist in computational biology at Celerity in Boston. She did a lot of the computational work, trying to understand what these cells are doing in our single cell library. So in this experiment, we took 3 control brains. We took 3 brains with brain metastasis. We sorted out the myeloid cells as well as the cancer cells, but I won't talk much about them. And we looked to see what are these cells doing? What kind of cells are they and what are they doing? Already when we did our clustering analysis, you can see this massive shift. So many of the myeloid cells in the brain from the metastatic mice were subclustering apart from the control brains. So we know that they have a different gene expression program. When we used gene scoring from bulk RNA signatures that have been published before, we found that these satellite clusters around the periphery here were actually clusters coming from the bone marrow. So bone marrow derived macrophages and monocytes, B cells, neutrophils. But largely these main clusters, so this big one here in particular that's making this big transcriptome shift, these are microglia and they score high for microglia expression, gene expression patterns. So we look to see what's different about the microglia from the metastatic brains and the control brains. We found 3,715 genes differentially expressed. We did GO-term analysis. Overwhelmingly, we found, in contrast to TAMS, previous published work about TAMS and glioma, microglia seem to display a pro-inflammatory signature. So they upregulate things like interferon response, cytokine production, antigen presentations, kind of the opposite of what we thought they were going to be doing. Again, looking at the actual genes, like I showed you in the other study, we definitely saw upregulation of lots of interferon response genes, tons of chemokines and cytokines, and lots of genes for antigen presentation. All these MHC1 and 2 genes, proteasome processing, CD74, just kind of what we expected from the GO-term analysis. And the expression of these genes was very specific to the metastatic microglia. You can see here how much higher the CD74 is in these cells that were from the metastatic brains. So very specific upregulation. A postdoc in the lab, along with Katrina, went to validate this at the protein level using flow cytometry. So we picked 3 of the top genes from the RNA sequencing data: CD74 and MHC2 for antigen presentation, and BST2, which is an interferon response gene. And in 4 different models of brain metastasis, we actually saw upregulation of all 3 of these markers in the microglia. So again, 1 human model, and now we're looking at 3 different mouse immune competent models in this study. We've also used the codex to do multiplex IF to look at the expression of these proteins in situ to see where they are actually being expressed, where the microglia are and what they're doing. And you can see here in this low magnification image, we have localized the tumor regions as well as the distal regions of the microglia. And TMEM being a marker for microglia, you can see in the distal regions, they kind of maintain their normal morphology. Whereas in the tumor regions, you can see the microglia starting to accumulate around these green GFP positive cells here. When we looked at MHC2, you could see some of the cells within the tumor regions, the microglia starting to upregulate MHC2 like we saw by flow cytometry. And furthermore, the interferon response gene, a different one this time that works for immunofluorescence, we saw also upregulated in microglia, although in overlapping, but not exactly the same subsets of microglia. So that we thought is interesting and something that we'll get back to in just a minute. So we're able to localize these, validate this in situ, and also look further to see that really these cells are co-localizing with the tumor. They're not expressed. These markers are not expressed in distal regions. So then we wanted to know functionally, are these cells actually tumor suppressive, or are they just upregulating these genes and proteins? So we turned to a genetically engineered mouse model that actually lacks microglia. We got these great mice from a lab out in Cambridge, and we asked whether, do mice without microglia have more metastasis? Really cool model. It's based on this deletion of this enhancer region for CSF1 receptor. I don't have time to go into it, but it's been published that in fact, these animals lack microglia, but they maintain other types of macrophages and monocytes from the periphery. So basically in this experiment, what we did was we injected the breast cancer cells, EO771 cells, into the mice. And then we harvested them basically at day 14 to look at tumor burden essentially in wild type animals that have microglia versus knockout animals that lack microglia. Already by day 7, we started to see these mice showing overt clinical signs of bad disease, the knockout animals without the microglia. And at endpoint, when we looked at these animals more carefully, we noticed something interesting. So not only was the burden higher in these knockout mice, but actually we saw that some of the wild type animals were actually able to reject the metastatic cells, the tumors that were outgrowing, whereas in the knockout mice, we just didn't see ever tumor rejection. They just kept growing. And so what do you think of when you think of tumor rejection, right? Well, we thought about T cells. We thought, well, maybe the microglia are promoting adaptive immunity. They are macrophages, right? It's not well accepted that microglia can do this function, but we thought maybe they're actually supporting the T cell response in some way, either, again, from our RNA sequencing data through antigen presentation or some other supportive functions. And I got to wrap up in a couple minutes here, but basically when we went back to our fire wild type mice that have microglia versus our knockout animals that don't, we injected the cells and we looked at the T cell and NK cell response at day 7. So like kind of midway in tumor progression. And in fact, we saw that the knockout animals without microglia had a dramatically attenuated NK cell and NKT cell response. We just didn't see many of them. And furthermore, the same for the CD8 and CD4 T cells. Even the Tregs also, we saw fewer of these cells essentially in the mice lacking microglia. So this led us to believe that for some reason, either when you don't have microglia, you either don't get as much infiltration or you don't get as much expansion locally once the cells are getting in. So we then looked to see whether these T cells are actually controlling brain metastasis. We did this experiment where we looked at tumor outgrowth in animals that either have been treated with something that prevents T cells from trafficking to the brain, or in knockout animals that just don't have T cells altogether. As expected, we saw fewer T cells in the treated and the knockout mice. And we also saw that these animals were more like our knockout animals, where they just were not able to basically reject tumors, whereas our vehicle treated animals rejected tumors in sort of the same frequency that we'd observed before. And furthermore, we had more burden. So not surprisingly, when you don't have T cells, the tumors can grow better. So what we concluded from this was that our microglia are essentially not sufficient to control brain metastasis without the T cells. So they're probably doing their anti-tumor effect through the T cells in some way. So then we started to look at marker expression microglia from mice lacking T cells. And the interesting thing that we found was in the RAG and the treated mice that don't have T cells, the microglia just never really upregulate as much CD74, so as much antigen presentation machinery. And instead, they express lots of interferon response, suggesting that T cell infiltration is critical for this antigen presentation response by microglia. Again, not that crazy, like we know in the periphery that macrophages and T cells sort of cross-activate each other. So we started to look into that a little bit more, whether this cross-activation, reciprocal activation is important for the anti-tumor response. And these last experiments were basically done by Aaron and others in the lab, essentially trying to recapitulate our single cell experiments, but now in a fully immune competent setting, so in a mouse model of brain metastasis rather than a human cell line. And what we did here was we looked at the immune response at day 4 as well as at day 10. So we did this time course in both wild type animals that have microglia and T cells, and then in the RAG knockout animals that don't have the T cells, again, to see whether the T cells are also are necessary to fully activate microglia to fully promote this anti-tumor response. When we did clustering, we saw a robust immune response in the brain, microglia, infiltrating monocytes, T cells, NK cells. And not surprisingly, what we found in our wild type animals was that the T cells increase a lot at day 10. You can see that here in salmon. And they don't increase in the RAG mice because the RAG mice don't have T cells as expected. So we see increases in both T cells and monocytes from day 4 to day 10. Knowing that, we went to look to see what the microglia are doing at day 4 and day 10. Here's clustering of only the microglia. And we found this subclustering pattern. And what we found was that at day 10, in the mice that actually have T cells, we see an expansion of the population of microglia that upregulate the antigen presentation machinery. And that in mice without T cells, in the rag mice, basically shown in this color here, they just don't ever upregulate fully the antigen presentation machinery. You can see that by CD74 and these MHC genes. So our antigen presentation seems to coincide with T cell infiltration, and no antigen response is observed without the knockout animals. When we looked at the T cells, we saw, in fact, that we got a much higher T cell. The T cells were becoming more activated at day 10. So we see more effector T cells and fewer naive T cells. Again, suggesting that as the T cells are coming into the brain, they're activating the microglia, and the two of them are working together, essentially to reject the tumors in these models. And we found basically using a human data set that these genes associated with antigen presentation, so MHC2, actually were predictive of better prognosis in patients, suggesting that people who have this anti-tumor response acting are actually probably doing better. And then finally, what I wanted to leave you with is essentially a sort of our conclusion to all of this. So what we know so far is that it seems that the brain can mount a productive anti-tumor response against brain metastasis. When you have microglia, more cytotoxic T cells, less Tregs. When you don't have microglia, you get more of an immune suppressive microenvironment, more monocytes, more Tregs. And when you don't have T cells, the microglia don't seem to become fully activated, and you get more tumor progression and more burden. One last thing that I wanted to point out was that what we're trying to figure out now is that we know that the microglia and the T cells can mount an anti-tumor response. They just don't do it fully effectively. Some of these animals are still, the tumors are still growing. So we're trying to figure out how we can boost this function of these two cell types in the brain. And so what we've been thinking about is, for example, some of these antigen presentation drugs like CD40 agonism, for example. And this work was done by Tim, who's actually in the audience, as well as Aaron, in actually a rotation project, I think, just to see whether if we can boost antigen presentation by microglia in the brain, can we get more anti-tumor T cells? And in this preliminary experiment, it does seem with CD40 injection, we get a lot more CD4s and CD8s in the brain. And so I'm going to just leave you guys with that. That's kind of where we're at with that study, hoping that we can learn new ways to improve anti-tumor immunity in the brain. And these are all the guys that I want to thank for the project. I mentioned most of them already, so I won't mention them again. But I want to just thank everyone for all their hard work. Many of them have already gone on to greener pastures since this time, as well as all of our funding. And if you thought you were going to get away in this talk without me showing old pictures, you were wrong. So these are, I had a nice trip down memory lane last night looking through all of our old pictures. And the ones that really pop out are the pharmacology parties over the years, which you can see here, Owen and Jamie having a blast dancing here. 2005, I think the whole lab here in 2007. I don't know what we were thinking with the Santa costumes in 2006. But do you guys still have these parties? I was just thinking, like, I don't know if my liver would have made it another 10 years with these parties, but I will never forget these pharmacology parties. So I hope you guys are enjoying every minute of graduate school here at UCLA. And then just a few more pictures of graduation. Andrew, you're in a lot of these. Owen, after the thesis defense party. It really was quite an adventure. And for those of you grad students in the audience, you know, I've had a blast since, but grad school is a special time in life, right? It's like I said, you kind of grow up in grad school. And so I hope you guys are enjoying the journey. You know, it's all about the journey and not about the destination. So with that, I'll go ahead and stop and see if you guys have any questions.
Excellent. Thank you so much, Devon. I'll ask the first question. So a lot of what you were talking about is these very early metastatic cells. And so I'm just curious, have you looked at later time points? Are the phenotypes conserved, or do they start to then become more like the primary tumor?
That's a great question, actually, and questions asked by many of our reviewers. So in the first paper where we did the qPCR, we actually were able to look at advanced metastasis too, and they do look more like primary tumor. And so I think, you know, in all the history of people doing RNA sequencing, looking at primary tumors and metastatic tumors, they kind of look the same, because they're very advanced. But when you do look at this seeding stage, they have this different phenotype, which is, is not surprising, but it was interesting to see what it was. In the RNA sequencing experiments, we haven't looked at more advanced meds yet. That's a really good question. Oh, the box.
Like, this is a microphone. I don't. So in the experiment in which you poisoned complex 1
Yes.
irreversibly, to create more ROS,
Yep.
which created more metastases. Did you harvest those metastases to see if OXPHOS had kind of returned to those?
That's a really good question. No, usually because it's an in vitro culture where we treat them in vitro, because treating the mice, not good.
Right,
Like you
right.
can't do that to the mice, right? So we do a pre-treatment in culture, and then we know for sure after like 36 hours in vitro, after the treatment, they return to their native metabolic state. So it's really a very transient change in ROS and also in OXPHOS and the OXPHOS treatment experiments that seems to make this effect. And it's not that the cells die. We looked at that too. They survive. They bounce back. They make primary tumors. But something about that, like, I don't know, 2-3 day window of being attenuated for OXPHOS or producing more ROS, makes them, it's probably like that initial seeding step, right? Because seeding usually happens within the first 24 hours is what most people think. Cells don't survive and circulate. You inject them, they either go and seed or they die. And so probably we're affecting that phase. And that they either want more ROS or more OXPHOS in that window of the metastatic cascade. That's a good question though.
Thank you.
Yep.
So first of all, can I get copies of their pictures?
Sure.
Second, in the immune competent model, where you're getting clear effective T cells, but in concert with the microglia
Yeah.
and other cells. What's the nature of the antigen or antigen?
Oh God, I would love to know that. In fact, I was just talking to Ken about this study and like where to go. And, it's a new field for us, immunology, less so for you. And so we've been thinking about how to do those kinds of experiments. We have thought about how we could look, because we actually have yet to see for sure that microglia are presenting antigen. We have evidence that they're doing so by gene expression, but we've not done any of these, like peptide loading experiments where you can see whether they actually activate T cells. We're starting to do that. But regarding what the tumor antigen might be, we're not entirely sure. That's a good question. Yeah, we should.
Yeah, I was curious. Metformin isn't necessarily a clean inhibitor, but I'm curious, do patients who are on metformin have differences in metastasis?
That's a great question. And it's very convoluted, the data. So there's epidemiological data that show that patients on metformin, because they're diabetic, have less metastasis, but more cancer. It's weird. And there's been a lot of that, because I think there were these trials where they were giving, antioxidants to patients to see whether they would get less cancer. And it was actually quite the opposite. And so they had to take them off these trials. They had to stop the trials early. And so that made the field, in my understanding, basically think that this is not a useful approach. But it turns out that tumor initiation and metastasis rely on different things. And so it may be that antioxidants and OXPHOS are not relevant at the tumor initiation stage, or even the alternative. But in terms of metastasis, once you already have cancer, that metformin might be useful. I don't know, honestly, whether those experiments or whether those kinds of trials it's controversial for sure, but a really good question, because there are these drugs out there.
So just in a matter of time, is one question online, so maybe we can do a quick answer. I think this is probably related to that upregulated lipid metabolite that you had shown.
Yes,
something
yes.
I was asking about
In the metabolomics, the steady state.
asking about polyunsaturated fatty acids as a nutritional approach. Have you thought about that?
Oh, yeah, actually, that's a good. We haven't thought a lot about it, but we have a collaborator who he's a new faculty at UCI, who we're starting to think about, dietary intervention, maybe more like they're in a therapeutic way. But yeah, that's a good question, whether you could alter like fatty acid uptake and then oxphos as a byproduct. That's a good question. Yeah.
Excellent.
Yeah.
All right. Thank you so much.
Well, thank you all so much. That was fun.
Nice.